national professor and a former DDG. He comes with 30 years of experience, 30 plus years of experience in the rice. So he is going to present on genomic revolution, discovery and deployment of broad spectrum blotch resistance genes in rice. So over to you, sir.
Okay, good morning to all. Very good morning. I would like to welcome you all for this presentation, specifically Honorable Vice Chancellor Dr. Patil, who spared some time to join here. Can you show my slides? So it's a very interesting symposium, and I would like to thank the organizer, particularly Dr. Sundaram and also Dr. N. Krishnan Kumar, our former DDG, for inviting me here to deliver this talk. Actually, this is a work on various aspects of plant genomics and blast resistance, on which I'm working for the past more than 35 years. And I would like to tell you how we can approach a particular problem and then try to solve that problem systematically over the years. This will also give you an idea. If you spend your resources vertically, then you are bound to get publications, products, patents, and partnership. So I'm sure that they are going to make efforts to show my slides, but by that time I would like to tell the students that it's very important nowadays to select a particular researchable issue which has national and international significance, and then go on chasing your dreams by applying all latest approaches. Thank you very much. We can see now. It's there. Can you go to the first slide? So maybe I will start without the slides. My first part of presentation is on genomic revolution, in which I'm going to tell you how we started decoding of different genomes of eukaryotic species in India. And in that context, most of the students might be aware that the real molecular biology was started with the decoding of structure of DNA by Watson and Crick in 1953 at Cold Spring Harbor Laboratory, which laid the foundation of today's molecular biology. So that's what I was interested to tell you. And how can I move slides? It's not going. It's okay. So this is what I was interested to tell, and it will cover on genomic sequencing efforts, rice blast, its identification of gene, map-based cloning, and then mining orthologs and how we can deploy these genes. Can you go to the next? So in 1953, DNA's double helical structure was decoded by Watson and Crick. And the first sequencing efforts of DNA was made in 1976, that was RNA, and in 1977 by Fred Sangers. And you know that he was the scientist who got 2 Nobel Prizes, one on developing the techniques to decode double helical structure of DNA and also protein sequencing. And if you see the down below here, the first crop plant genome was sequenced in 2005, and in 2001 human genome was sequenced. All these genomes now available in public domain. Next slide, please. And this is how we can sequence a particular genome. Till 2005, we were using a typical Sanger's dideoxy method of sequencing, but after 2005, pyrosequencing was developed, and now we have Illumina, PacBio, and Nanopore-like platform where you can sequence the genomes. And I would like to tell you here, in 1994, when I manually sequenced a genome, within 7 weeks' time, we could get only 100 nucleotides. Now, in 7 hours, you can get full genome. Next slide, please. This is one of the most important efforts in which India was participating in international efforts to sequence rice genome, and we were sequencing chromosome number 11 of rice. And that's how we sequenced Nippon Bear variety of rice with 388 genome size and predicted 37,500 genes in this particular variety. Next slide, please. Subsequent to that, another international genome sequencing effort was there in which India participated and sequenced chromosome number 5 of tomato. And here we sequenced cultivar Heinz 1706 with 900 million base pair genome size and predicted around 34,000 genes in tomato. Next slide, please. Then India also sequenced independently pigeon pea genome, which was published in 2012, and here we used only next generation sequencing approach to decode the complete genome of pigeon pea variety Asha. Next slide. After that, if you see here, these are our efforts in which we sequenced mango, jute cultivar, wheat cultivar C306, lathyrus, wild rices, black rice of Assam, guar, and tea. And all these reports are published, and in these all genomes, we identified many agriculturally important genes and also genes related to disease resistance. Next slide. Besides that, many plant pathogen genomes were also decoded by our group, like Puccinia genome of all 3 Puccinia species, and then Magnaporthe and Fusarium oxysporum. Here, we identified gene for pathogenicity, A virulence, and different effector proteins. Next slide, please. The most important slide which I would like to show here, in 2000 for sequencing human genome, we need around $100 million US, but now in 2022 and 23, within $100 US you can sequence complete human genome. Till 2000, we were having few genome sequences available in public domain. If you see in plant and animal species, there were only 2 species were available in public domain, but now more than 30,000 species genomes are available in public domain. Next slide. So utilizing genome sequence information, if we see here, after the decoding of genomes of different species, most of the disease resistance genes were decoded in Arabidopsis. Before 2002, when genome was not available in public domain, only 14 genes for disease resistance were cloned. But after 2000, more than 31 genes have already been cloned. Similarly, in rice, only 4 genes were cloned before 2002, and 24 genes were cloned after 2002. So that shows how genomic information is important for cloning and characterization of different genes. Next slide, please. And using genomic resources and genomic sequences, these are the genes which we decoded in our lab, starting from PI54, PI54RH, PI54OF, and then many promoters, and also genes related for programmed cell death, and also and A virulence genes for PI54. Next slide, please. So I would like to concentrate here more on telling you the story of cloning and characterization of PI54 genes, how we started this work. And you know, Dr. Prasad just now presented how this disease is important nationally and internationally. Next slide, please. If you see internationally, this disease is present everywhere where rice is grown, and in India, even all parts of the country, you can have this disease already presented by Dr. Prasad. And it's responsible for approximately 30% rice production losses, which can feed 60 million people every year. We estimated that for the management of rice blast, around 68 crore rupees is being spent in basmati growing areas of India. And the best management option is host resistance, for which not only you need sources of resistance, but also need gene for resistance. But by using conventional method, which is very time consuming, laborious, and amenable to linkage drag, we have to concentrate on a specific gene. Next slide, please. So our first question in 1996 was, is there any rice line resistant to rice blast in India, which was present? How many genes control that resistance? And then third question was how to discover that gene. Next slide, please. So for doing that particular experiment, we exposed 13 rice lines which were known as differential host to 119 strains of Magnoporthia fungus, which was collected from J&K, Himachal Pradesh, and Uttarakhand. And we identified a line TTIP. You can see this particular red arrow, which I showed here. It was completely resistant to 119 strains of Magnoporthia raceae. And this is the phenotype of TTIP leaf. And to identify a gene in this particular variety, which contains 36,000 genes, you have to develop a mapping population. For doing that, you have to cross this with a susceptible line. So we crossed TTIP with HP2216 and developed a mapping population. Next slide, please. So we developed a linkage map here, and you can see from this green line here, PI54 was mapped with two markers, RM207 and RM224, with 0.7 and 10.7 centimorgan distance. And when we looked at the literature, there was no DNA markers available with us. So we found the sequence of rice genome from the databases and designed 40 new SSR markers in the Nipponware genome and used those 40 markers for genotyping of our mapping population 208 plants and developed, next slide please, and developed a typical fine map of this particular gene here. You can see the fine map which we developed by using 40 new SSR marker. Out of those 40s, only two markers, that is TRS26 and TRS33, was present at 0.7 and 0.5 centimorgan distance. So in terms of centimorgan, this is a very short distance, but in terms of physical distance, it comes around 142 kilobases. So to identify one gene in 142 kilobases, again, it's a big task. To do that, we used various computational methods and identified 18 genes within these 142 kilobases, and all those 18 genes were annotated. And finally, we found that there was one gene which we named as a candidate gene, which was showing similarity to resistance-like genes. So we named it as a candidate genes. So starting from a complete genome of rice in TTIP, with all genetic and genomic approaches, we narrowed down to a small fragment of 1.5 kV as a candidate gene. Next slide, please. So to clone this particular gene from resistant line TTIP, we amplified this from both resistant and susceptible line. You can see P1 and P2. Again, sequenced this particular fragment, and we were happy to see that we found a typical gene structure which has open reading frame, promoter sequences, and poly A tails. When we compared TTIP sequence with the Nipponware, which is susceptible to rice blast, in the susceptible
allele of PI54, there were 3 insertions. One was in open reading frame, another was in promoter region, and third one was in poly A tail. And when this typical protein structure was designed for PI54, we found a horseshoe-shaped structure. You can see here a protein structure here, which is red in color, which is called leucine-rich repeat region, LRR. And this is the region which is very, very important to receive signal from the pathogen side. Next slide, please. So to prove the function of this particular gene, this gene was cloned along with its native promoter in a plant transformation vector. So if you want to prove the function of a gene, you have to put this gene back in a susceptible line. For doing that, this gene was transferred in PI54 along with PI54 promoter regions and transferred to the susceptible line of rice type I309, and using all these transgenic methods, when we analyzed those plants with PCR, we found that all those plants were PCR positive, and our southern analysis tells us this, that most of these plants contain single copy insertions. When we did expression analysis of all these transgenic lines, we found that the gene is expressing similar to that of its native allele, which is present in TTUP. And our final test came when we inoculated those lines with virulent strains of Magnaporthia oryzae. You can see that all the transgenic plants are resistant to that particular strain. The next step for validating this particular function was using RNAi approach, which we did, and we knocked it down. And we also found that the gene which we cloned was showing resistance to the strains, at least 4 strains collected from North India, East India, and also South India, which tells us that PI54 is a broad spectrum resistance. Next slide. So next question was, our question was how this single gene is controlling resistance phenotype in rice. And to do that, we performed global gene expression analysis. Next slide. And we showed here with microarray analysis where in RNA of our transgenic line was hybridized with gene chip, which contains 56,000 genes. And our analysis shows that all these red color indicates on right-hand side is that all those genes related to response to stimulus, signal transduction, transcription regulators, programmed cell death, etc., they are expressing in our plant, which is resistant to Magnaporthia oryzae. Next slide. And our differential gene expression analysis tells us very clearly that in transgenic lines, 107 genes exclusively upregulated, and those genes were mostly related to transcription factors, programmed cell death, and signal transduction. These genes were not only regulated or expressed, but also producing enzymes like peroxidase, polyphenol oxidase, and beta-glucanase and chitinases, which are considered as defense response related genes, and these were present in transgenic lines. Next slide. We then developed a typical molecular model how this gene is working in a typical plant cell here. You can see here the gene is shown here. Unfortunately, this pointer is not working. But you can see this is a typical rice cell which is infected by Magnaporthia spores. And when plant infects rice cell, it releases certain proteins which are called effector molecules. These effector molecules are recognized by resistant gene PI54 at leucine repeat region, and then all these dotted lines which I showed here, it shows that it regulates the various defense response related genes within a plant cell. And then finally we concluded that PI54 regulate the regulators of defense response against rice blast pathogen. Next slide. And then we also showed in our subsequent publications that PI54 was the smallest gene cloned from literature, and it contains a typical zinc finger domain which was not present in rest of the genes cloned from different plant species, that makes it a unique genes cloned from rice. Next level. And our proteome analysis also tells us that by iTrack-based proteome revealed that PI54 regulates secondary metabolism hormone signaling, and pathogens-related proteins. And that's how you can say that this particular gene is working within a rice cell. Next slide. So next question was, since gene we have cloned here is a broad spectrum genes, then our question was how its alleles are working in land races and wild species of rice. Next slide. We collected 92 land races from different parts of India and then looked at various features of this particular alleles, particularly nucleotide binding sites and leucine repeat region, and we developed even allele-specific markers based on two conserved region of 163 base pair and 144 base pair and developed allele-specific DNA markers. And then finally, with this analysis, we concluded that PI54 allele in rice land races may offer broad spectrum and durable resistance, making them invaluable for breeding programs. Next slide, please. To mine its orthologue in different wild species, rice has 24 wild species, and one of them is Oryza rhizomatis. And you can see here, this is a work of one PhD student. He mined this particular allele, Oryza rhizomatis, and then compared this particular allele with TTUP alleles and Nippon bear alleles, and finally validated its function, and we concluded in our F figure here that the allele orthologue which we cloned from Oryza rhizomatis providing resistance to more than 6 strains of Magnaporthia oryzae. The paper was published, and also we got patent on this particular orthologues. Next slide. The another orthologues which we cloned is PI54OF from another Oryza officinalis, a wild species which was cloned by Dr. Devanna, who is student of this university and now working at senior scientist at CRRI Cuttack and also sitting here in the audiences. He made a very typical analysis of this orthologue cloned from Oryza officinalis compared with all the alleles which we cloned from Oryza rhizomatis as well as TTUP and susceptible lines, and then filed a patent on this particular orthologues. Next slide, please. And showed its function in both Indica and Japonica type of rice, and very clearly we showed that PIOF is providing high level of resistance to multiple strains of Magnaporthia oryzae. Next slide. Now, next question was, if this particular allele and orthologues which we cloned from TTUP, Oryza rhizomatis, and Oryza officinalis, where these are expressing in a protein gene, either it is present in DNA, in the nucleus, or in the cytoplasm, or cell membrane. So where it is present, therefore doing that analysis, one of my PhD students, she did this analysis and found that and showed very typical localization of PI54 and PI54RH. These are localized in the host cell membrane, whereas PI54OF is localized in the cytoplasm. And we showed that this diversity is a boon for breeding program because of their multiple expression in different compartments of the cells. Next slide, please. Now, when we cloned this particular gene, the next step was how to utilize this gene. And for doing that, introgression of PI54 in popular rice varieties was started in 2008, and we looked at their performance in different agronomical backgrounds and different zones of the country. Next slide. And what we did here, you can see here, but what is the material we provided to the breeders? We were providing the gene, we provided gene-linked markers, TRS26 and TRS33, as well as gene-based markers. Not only that, also donor important lines, TTUP as well as a TTUP-derived derivatives, recombinant inbred line 4, which we provided to the breeders in 2008 in a network program. Next slide, please. And this was the strategy which we adopted here. You can see an example of Pusa basmati 1, which is highly susceptible to Magnaporthia oryzae, and the TTUP phenotype, it was crossed with Pusa basmati, and then the subsequent segregating populations were monitored by using gene-linked and gene-based markers, and wherever this gene was present. You can see the band was present, and also we also did phenotyping at Indian Institute of Rice Research as well as at other places to look at the segregating lines and their phenotypes to Magnaporthia oryzae. Next slide. And those lines were evaluated in different parts of the country. This is one of the examples of Karnal, Pusa, Bihar, and then Panipat and IARI field, and this is the monitoring team which are evaluating those lines which contains PI54. Next slide. And also similar to that, this is an example of Pusa Samba 1850. This is a variety which was released by IARI group, and then Pusa Basli 1885 again contains PI54 along with other genes, and then HPR 2880 which was evaluated at Palampur and released in Himachal Pradesh, and then Shalimar rice 6 containing PI54 was released by our colleague from Sher-e Kashmir University at Kashmir. Next one. And similarly, our colleagues, Dr. Sundaram's group, this is an example of DRR Than 62, which contain PI54 along with other genes, and also at Tamil Nadu, CO51, which contain PI54, and the phenotype of this particular line. Next slide. So when we looked at literature this year, and this hard work was done by Dr. Devanna, we found that more than this gene was transferred in more than 40 improved varieties of rice and 22 advanced breeding lines. And this is the pan distribution of PI54 in India. Wherever rice is grown, all those 40 varieties are listed on that side which contain PI54, and these are the developer institutions including IRI and also Indian Institute of Rice Research, particularly Dr. Sundaram's group, and also one report from Dharwad where they transferred this particular variety, this gene in N10 variety. Dr. Prashanti, she did this work, and all these colleagues now made this particular gene which is present in almost all lines grown in the country. This is the example only from public institutions, and I'm sure that many private institutions, they have transferred this particular gene in their varieties. Next slide, please. So finally, to conclude this particular presentation here, these are the milestones of PI54 discovery here. We started in 1996 on characterization of Magnaporthia populations, identified TETUP in 2002, then a gene was mapped on in 2005, and then again cloned in 2005. In 2008, this MAS program was started. In 2011, this gene was functionally validated. In 2012, a molecular model was developed, and also in 2012, we cloned its ortholog from Rhizomatys. In 2014, another ortholog was cloned from Officinalis. In 2015, alleles were mined from 96 rice lines. In 2016, AVR PF54 was cloned, and in 2025, if you see here, the gene is transferred now in more than 40 rice lines. And as I told you, the PI54 associated studies gave us more than 70 publications, 5 patents, 3 products in the form of varieties, and more than 22 PhD students were trained on this particular gene, and it helped in developing human resource in molecular biology in the country. Next slide, please. So to conclude my presentation here, we sequenced genomes of rice, tomato, pigeon pea, wheat, jute, mango, guar, black, Lathyrus, and many plant pathogens, and this sequence is available in public domain. We also cloned PI54 blast-resistant genes and its orthologs from wild species, developed gene-based markers, and now this new gene or novel gene which we cloned, PI54, has already been transferred in more than 40 varieties of rice, creating a silent revolution in rice breeding. Next slide. These are my all students at that time when we completed, and these 22 students which are listed here, they are exclusively worked on PI54 gene, and I will take only 1 minute to explain here, because most of the students are working here, but where these students are present here. The Prem was the first MSc student who developed first cross in 1999 in my lab, followed by Vinay, who did RFLP analysis for mapping of this gene, who is now principal scientist at Indian Institute of Wheat and Varley Research Institute. And then Madhav cloned this particular gene in 2005 in my lab. He is director of Tobacco Research Institute, Rajamundry. Satyapal did functional validation of this particular gene, who is now in Indian Revenue Services and working in Japan. Santosh did this microarray analysis and transcriptome analysis, and he is senior scientist at Indian Institute of Millet Research. And then Archana did allele mining, and she is principal scientist at BARC, Bombay. Alok cloned orthologous of this particular gene from Oryza rhizomatis, who is senior scientist at Indian Institute of Pulses Research. Donald worked on functional validation of OF, and he then shifted to Indian Institute of Science, Bangalore, for doing his PhD. Amit was involved in doing functional validation of PI54, who is running his own company in Bihar. Sangeeta was involved in decoding the genome of this rice blast for identifying defense response gene and resistance gene. Shoham cloned this AVR PI54 gene, who is scientist at Indian Agriculture Research Institute, now New Delhi. Shalu did allele mining, she doing postdoctorate from US. Pankaj did allele mining of AVR gene, who is now associate professor. Devanna did allele mining of orthologous genes from Oryza officinalis, who is senior scientist at Indian Institute of CRRI, Cuttack. Joshita did promoter cloning, and she is senior scientist at IARI, New Delhi. Kirti was involved in doing work on RNAi, and she is now a scientist in forensic lab in New Delhi. Mandeep did proteome analysis, and she is now postdoctorate in US. Priyanka was involved in transcriptome analysis of PI54. She is now associate professor at MIT University. Jyoti did expression analysis again in PI54, and she is doing postdoctorate from Israel. And then Sarkar was doing computational analysis of different alleles. He is a professor at Central University of Assam. Banita did again this expression analysis of PI54. She is now associate professor in Himachal Pradesh. And Ritu cloned pathogen-inducible promoter. She is doing postdoctorate in US. And this is how we collaborated with scientists from IARI, HPKBB Palampur, and also CRRI Cuttack, and also Dr. M.S. Prasad from Hyderabad. And most of the funding from this whole work came from ICAR, DVT, World Bank Project, and also Department of Science and Technology, Government of India. So to explain this to the student, those who are sitting here, if you invest your mind and also do more critical science, I think there are a lot of opportunities in science also, and basic sciences also, wherein you can do a lot of work and make your career in sciences. So thank you very much, and we can have some questions if you wish.
Thank you, Dr. T.R. Sharma, for amazing journey of how field lab discovery can take it to the field transformation and bringing a lot of impact in the rice field. Thank you. Nice presentation. I think we have one question. We have time left for only one question.
There is no question. Thank you. So you can send your question to me by email if you wish.
So now we'll break for the tea. I'll request for announcement.
Now we shall break for a very brief of 10 minutes tea break. Very short break of 10 minutes to have a tea and assemble back in the same hall for the next presentation from Dr. R.M. Sundaram, sir.
ಹರೀಶ್ ತೆಗಿ. ಇದೆ ಸರ್ ಸರ್ ಈಗ ಬೇರೆ ಹಾಕೇನು ಸರ್ ವರ್ಕ್ ಆಗ್ತದೆ ಸರ್ ಅದು ವರ್ಕ್ ಆಗಲಿಲ್ಲ ಸರ್ ಬೇರೆ ಹಾಕಿದ್ರೆ work ಆಗ್ತದೆ. ಅದೊಂದು ಪಾಯಿಂಟ್ ಮುಂದೆ ಸರ್ಸಣ ಬೇಕಾದ್ರೆ ಬಿಡಿ ಸರ್ ಹಾಂ
ಸ್ವಲ್ಪ ನೋಡಕ್ಕೆ ಅನುಕೂಲ ಆಗ್ತದೆ ಇಲ್ಲ ಅಂದ್ರೆ
ಹಾಂ ಇಷ್ಟೇ ಸಾಕು ತೋರಿ ಸರ್ ಇಷ್ಟೇ ಸಾಕು ಈಗ ನೋಡಿ ಸರ್ ವರ್ಕ್ ಆಗ್ತದೆ ಸರ್ ಸರ್ ಬ್ಯಾಡ್ ತೋರಿ ಸರ್ ಈಗ ವರ್ಕ್ ಆಗ್ತದೆ ಸುಳ್ಳೆ ಈಗ ಯಾಕಂದ್ರೆ ಸರ್ ಇದಂತೂ ವರ್ಕಿಂಗ್ ಇಲ್ಲ ಸರ್ ಇದ್ರಾಗ ಈಗ ಮನೆ ರಿಮೋಟ್ ಅದಕ್ಕೆ ಯೂಸ್ ಮಾಡ್ಕೊಂಬಿಟ್ರಿ ಸರ್ ಇಂಥದ್ದೊಂದು ತರಿಸಿ ಇಟ್ಕೊಡಿ ಸರ್ ನಾಳೆ ಯಾವ್ದು ಕೊಟ್ಟಿದ್ದು ನಾನು ಇದು ಕೊಟ್ಟರೆ ಸರ್ ಎರಡು ಮಿಕ್ಸ್ ಅದು ವರ್ಕಿಂಗ್ ಇದೆ ಸರ್
ರೈಸ್ ಕೋಆರ್ಡಿನೇಟರ್ ನಮಗೆ
ಹಾಂ ಈಗ ವರ್ಕ್ ಆಗಿದೆ ಸರ್
ವರ್ಕ್ ಆಗೈತಲ್ಲ ಇಲ್ವಾ
ಶಾಲರಿ ಚೇಂಜ್ ಮಾಡಿದ್ವಿ ಸರ್
ಸರ್ ವಾಟ್ಸ್ ಪ್ರಾಬ್ಲಮ್ ಆಗೈತಲ್ಲ
ಹೌದು
ಸೆಲ್ ಪ್ರಾಬ್ಲಮ್
ಶಾಲೆ ಸರ್
ನಾಟ್ ಕಂಪ್ಯಾಟಿಬಿಲಿಟಿ ಪ್ರಾಬ್ಲಮ್ ಅಲ್ಲ
ನಮ್ದು ಏನು ಪ್ರಾಬ್ಲಮ್ ಇಲ್ಲ ಸರ್
ಅಲ್ಲ ಅವ್ರು ನೋಡ್ರಿ ಈ ಕಡೆ ಅಲ್ಲ ಈ ಕಡೆ ಮಾಡಿದ್ರೂ ಆಗುತ್ತೆ ಎಲ್ಲಿ ಮಾಡಿದ್ರೂ ಆಗುತ್ತಲ್ಲ
ಆಗತ್ತೆ ಸರ್ ಶಾಲೆ ಡೌನ್ ಇತ್ತು
ಇದು ಪಾಯಿಂಟರ್ ಇದೇನಿದೆ
ಪಾಯಿಂಟರ್ ಸರ್ ಅದು ಪಾಯಿಂಟರ್ ಯಾಕೆ ವರ್ಕ್ ಆಗ್ತಾ ಇದೆ ಪಾಯಿಂಟರ್ ವರ್ಕ್
ಇಲ್ಲ ಆಗ್ತಿದೆ
ಅದ ಸರ್ ಅಲ್ಲಿ ಕಾಣಂಗೇ ಇಲ್ಲ ಸರ್
ಮತ್ತೆ ಅಲ್ಲಿ ಕಾಣಿರೋದು
ಅದಕ್ಕೆ ಅವ್ರು
ಇಲ್ಲಿ ನೋಡ್ರಿ ಇಲ್ಲಿ ಕಾಣ ಇಲ್ಲಿ ನೋಡ್ರಿ ಮೈಕು
ಹಾಂ
ಬಟ್ ಅಲ್ಲಿ ಬರ್ತಾ ಇಲ್ಲ ಅದು ಅಲ್ಲಿ ಬರಲ್ಲ ಇರ್ಲಿ ಬಿಡು ಬಟ್ ದಿಸ್ ಇಸ್ ಆಪ್ರೇಟಿಂಗ್ ಅಲ್ಲ
ಆಪ್ರೇಟಿಂಗ್ ಆದ ಸರ್ ಇದು ಆಪ್ರೇಟಿಂಗ್ ಆದ ಸರ್ ಹೊಸದು ಕೊಟ್ಟಿದ್ರಿ ಅದನ್ನು ಹಾಕೇನು ಸರ ಇದೊಂದು ಇಟ್ಕೊಂಡಿರ್ತೇನೆ ಸರ್ ಸರ್ ಇನ್ನೊಂದು ಸ್ವಲ್ಪ ಮುಂದೆ ಮಾಡೋದಿಲ್ಲ ಬ್ಯಾಬ್ರಿ ಸರ್ ಸಾಕ್ಬಿಡ್ರಿ ಸರ್ ಬರೀ ಅವನೇ ಹೇಳ್ತಾನೆ ಫೋಟೋ ಬರಲ್ಲ ರೀ ಹಂಗೆ ಆಗಲ್ರಿ ಹಿಂಗೆ ಆಗಲ್ರಿ
Welcome back. Before we move to the presentations, we have an announcement here. There is a special interaction session for the benefit of students. At 6 o'clock, we have an event of interaction between the industry personnel and the students at 6 o'clock. I request all the postgraduate students of the university to kindly be present in the hall at 6 o'clock and actively involved in this interaction with the private sector and industry people present here. And also I request industry representatives, please make yourself available for the interaction at 6 o'clock in the evening. One more request from the organizing committee that all the registered participants should please carry the food coupons while you go for the lunch and dinner. Kindly carry your food coupons to the guest house to have a lunch and dinner.
Okay, welcome back. Now we have very important presentation here by Dr. R.M. Sundaram. I invite Dr. Sundaram, who is Director of Indian Institute of Rice Research, Hyderabad, and also I think one of the first developer of marker-assisted derived line using these for accessory genes, and did a lot of work on marker-assisted selection and gene mapping in rice for the past more than 25 years. So Dr. Sundaram, please.
Thank you. Honorable Vice Chancellor of this university, Dr. Patil, respected Chairman and Co-Chairman, and my dear colleagues, I'm going to talk about advances in molecular breeding and tools for multiple pest and disease resistance in rice. What I'm going to narrate is my last 20-year experience, which I've gathered for the last 28 years. And it's not something like a typical Bhagavad Gita or a sermon I would like to give. I would just like to share where we have succeeded, what we have learned in the last, say, 28 years in terms of breeding for multiple biotic stress resistance, and where we have failed, and how we sort of did a mid-course correction. So I'll be dividing my presentation into 5 parts. The first part, I'll give a brief introduction about the scenario of rice, which my colleagues have already explained. I'll also be talking about why biotic stresses are very, very important in the context of stabilizing rice production. Then I'll be talking about one of the most important steps in terms of deploying genes or identifying genes, that is molecular mapping. I'll talk only with a few case examples and talk about an important upcoming tool called association mapping. And I'll then be in the third part of my presentation talking about molecular breeding, what are all the various strategies, including backcross breeding, pedigree breeding, and also some of the new tools and techniques will be the fourth part of my presentation. In last 2 slides, I'll be summarizing what I have learned in the last, say, 25, 28 years in this particular field. So as you are all aware, we are #1 in terms of not only rice production, and we are slowly but steadily increasing our productivity also. And rice is very, very important. And we are also doing extremely well, not only in export of basmati rice, even in non-basmati rice also. So the scenario with regard to rice is looking very green, but it's not so green Because if you look into what we require by, say, 2050 or 2047, when our Honorable Prime Minister is expecting our country to become a developed country or Viksit Bharat, that's where the major problem is. Presently, rice is grown in about 47 to 50 million hectares, but by 2047 or 2050, the total rice area in which rice is grown is expected to decrease to about 35 to 37 or maximum 40 million hectares. We are going to lose outrightly around 7 to 10 million hectares of rice growing area. And there are other associated problems like biotic stresses, rapidly changing climate and all. So we need to work. And while breeders will be developing good varieties with higher yield, with better quality, we also need to protect the yield by protecting the varieties and hybrids against biotic stresses. So if you look into it, any survey says that in any given season, biotic stresses can reduce yields of rice up to about 30%. And what are all the biotic stresses which are of significant or important in rice? As far as insect pest goes, they are stem borer, leaf folder, gall midge, and then brown plant hopper. As far as diseases are concerned, we have blast complex, then bacterial blight, sheet blight, fall smut, and a few other minor diseases which have assumed major disease proportion. There is a reason why I have circled problems like stem borer, leaf folder, and also fall smut. This is principally because there is no known strong host plant resistance. So resistance breeding has not progressed really well, but there is some silver lining. My colleagues have done some seminal work in terms of identifying candidate genes or candidate loci associated with stem borer and fall smut. I'll narrate when I'll be talking about them. And finally, with regard to resistance gene breeding strategies, Dr. T.R. Sharma ji has very nicely, eloquently expressed it. While genes like PA54, XA21 are very useful, but it will always be better if they are deployed in combination, say PA54 with PA2 or PA9, XA21 with XA13 or this thing. Pyramiding or stacking multiple resistant genes is the way for us to move forward. And this is particularly accentuated because of a rapidly changing climate, wherein what we are seeing is we are seeing a transition from many of the minor diseases or pests to major pests or diseases. This is particularly true. This is a slide given by my colleague Dr. Padma Kumari. When Green Revolution started in our country, we had only 3 major insect pests. But as of now, we have got more than 20. The numbers are increasing steadily. And this is also true with disease scenario, which was ably illustrated by Dr. M.S. Prasad. When our country became independent, we had only one major disease problem, that is brown spot of rice. But as on today, it's 12, and the number is growing steadily. And one of the main reasons for this is that the climate change related vagaries like increasing temperature, shifting rainfalls, and shifting humidity patterns are actually accelerating or accentuating this particular problem in terms of making the pathogen or pest strains more virulent or more infective. And now coming to the second part of my presentation, I would just briefly give a glimpse about how we are doing molecular mapping, or how we are, before identifying and then deploying the genes or QTLs, one of the first and foremost tasks is to identify the genomic regions or the genetic loci associated with resistance, and to fine map them, and if possible, do a gene characterization, which was very ably illustrated by Dr. T.R. Sharma with respect to PR54 gene. And I'll be talking about how we work towards this particular task. So if you look into the flow with regard to development of resistant varieties, it starts with identification of resistant genes or QTLs in populations or in the land races or varieties, then identifying molecular mapping or marker development, then deploying those markers in marker-assisted selection, and then finally